Welcome to Data in Biotech, a podcast from CorrDyn where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. This week, we're excited to be joined by Jesse Johnson, founder of Merelogic, an independent consulting firm that helps early-stage biotech startups get their data under control so they can focus on the science. During this interview, Jesse highlights the unique challenges of working with biotech data, introduces the concept of a digital twin for the lab environment to better integrate data entry and experiment tracking, discusses the potential of machine learning to enhance efficiency in biotech research, and speculates on the role of large language models like GPT-3 in biotech as potential learning tools to assist in generating experiment protocols. Here we go.
Jesse Johnson, welcome to the Data in Biotech podcast.
Thanks for having me. I'm excited to be here.
Awesome. Just to kick us off, can you give everyone a brief introduction to you and your background?
Yeah, so I started off my career as an academic mathematician, did that for probably longer than I should have, and then transitioned to software engineering. I was a software engineer at Google for a couple of years, and then basically by following people that I worked with who had moved on to interesting other jobs, I followed them into kind of the biotech world. So my first biotech startup I joined about 4 years ago, a company called Celerity. And then after that, spent a couple of years at DuPont Therapeutics, both roles, so data science and engineering. So mostly software engineering, a little bit of data science. And then just in the last few months, I started as an independent consulting. So my goal is to help biotech startups get their data under control so they can focus on the science. That's the tagline.
Amazing. What was it like transitioning into biotech from your sort of deep math and software engineering background? I imagine there was some like culture changes and also a lot of terminology getting thrown around that it took a while to get up to speed on.
Yes, definitely. Yeah, the vocabulary takes a while. You have to be willing to ask stupid questions, which I'm willing to do. Not everyone always appreciates that. If you can find the people who are okay with that, you're good. Yeah, the culture, I really like the kind of the tech software engineering culture of leading by motivation and explaining why and things like that. I think in biotech, you get some of that. There's definitely leaders like that. There's also folks who have come from more academic labs where it's more about pumping as much as you can out of grad students and postdocs. And so there's a little bit of a culture clash of like, how much freedom do you want to give to people? How much do you need to just make them get things done? So it's been interesting, but it's an adjustment.
You made that transition several years ago now, so I feel like you're fully indoctrinated into it. But another question I had based on your background was just that you've been in both the bigger company and bigger pharma environment, but also smaller biotech and startup ecosystem. Why did you decide to focus on data problems of biotech startups?
Yeah, so a couple of reasons. I think the primary one is I really like that early stage research where it's very exploratory. There's a lot of flexibility. You're not weighed down by regulatory issues and things like that before. When the closer you get to clinical trials, the more of that there is, and the more focused you get, so less room for exploration. And so the biotech startups are pretty much just doing, especially those early stage ones, are pretty much just doing research. So that felt like the right place to do it. So that seemed like the right opportunity there. And then I like coming into a messy room and cleaning it up to where you can see, like the before and after picture versus coming into an already mostly clean room and cleaning it up some more. So I feel like the big pharma, they tend to have a lot of things in place where either they're good and it just needs to be maintained, or it's a mess and there are deep structural issues why it's always going to be a mess. And so I like being able to come in early on and get from kind of 0 to 1, as they say.
Mm-hmm. More problems of data exploration and like getting insights from the data, but also less existing infrastructure to compete with and trying to get the problem solved. Yeah, that makes a lot of sense. When you're starting to work with a biotech startup, what do they look like when you come in? When do they realize that they need you? How does that kind of start?
Yeah, so my thinking on that has actually evolved in these months since I started. So my primary focus has been very early stage startups where they're not yet ready to hire that VP of engineering or even maybe a CTO. My goal is to come in and help them build something that's very lightweight, low overhead, and we'll get them to, until they start getting that burst of data and they need to suddenly scale, I want them to be in a good shape to bring in a larger, more higher overhead tool that will get them through the next step. And so often I think that happens around the stage where most biotechs get to, where they say, okay, we've now done 3 or 4 assays on our compounds of interest. We want to see them all in one table, and for some reason we can't. It seems like it should be easy. And so I think that's where I can come in. The other type of project that I've seen more of as I've had more conversations is even platform-based companies or like farther on where they actually have an engineering team, they've put some effort into this. Often those engineering teams get so bogged down in the day-to-day, just trying to put out fires, keep up with the moving train, that there's often projects that they really want to do, that they know how to do, they just don't have the bandwidth to do it. And so there, I think I can often come in and do a strategic project that frees them up a little bit, just do enough of the project to where they can then take it over and run with it. So I'm exploring both of those types of projects right now.
Yeah. Do the startups that you're working with typically have a data team in place when you come in, or are you like standing up or helping them stand up their new data team that they're introducing to the organization?
Yeah, so for those early stage startups, I think they often will have computational biologists, data scientists, so folks who are really good at the analysis side of things and know how to work with data, but don't necessarily know the engineering side of it, the kind of the platform below that analysis, right? So they can do things on their laptop. Maybe they can upload things to an S3 bucket. There's a layer below of how do you organize that data? How do you build the infrastructure that makes sure that the next time someone comes in or someone leaves, that they can find the data, that they can rerun it. So making it reproducible and all of that, and making the data more of an asset that is value to the company versus just an operational kind of thing that just gets you to the next analysis.
Yeah, that makes sense. I imagine that there's this aha moment where they encounter some feeling of pain where they're just like, oh no, we're not going to be able to continue operating this way. We need to find someone who can help us overcome this. What do those problems look like where the teams start to realize that, oh, we need somebody like Jesse in here to help us get to the next phase of what our data maturity looks like?
I mean, my goal is to get in before it gets to that level of pain. But yeah, I think often one thing that I think most startups end up with is they want to see a single dashboard, a single view, a single table of all of their assets, whether it's compounds or biologics, and for each row, and then each column is an assay, and basically a decision-making tool that allows them to say, okay, which of these, if you look across all the data we've generated, which of the one is that we want to invest more in? What do we want to take to the next assay, next step? That seems to be fairly universal need, and often that's something that's surprisingly difficult to actually build internally.
Yeah, so there's that like portfolio prioritization problem that's at a level above the biology that's being done. And so the data that they have is in all these different places, and like that unified view is hard to get to. What makes that so hard?
I think often if you're doing analysis on a single assay, you're thinking about it in terms of, okay, I just want to get to an answer of what did this assay determine. Merging all of that together is surprisingly hard, especially if you're using different formats for different assays that work well, again, when you look at that one, but not the other. I think often data ends up either on people's hard drives or it's in a folder for that experiment, where if you know about that one experiment, you know where to find it. But if you're looking across multiple experiments, multiple assays, you don't know. And so I think there's a certain amount of kind of forethought that you need to put into how do we want to organize this in a way that someone who isn't deep in the science can come in and understand across all the different experiments and assays what's going on.
Yeah, so I'm assuming that these assays are happening on different pieces of equipment, right? And so the data that they're generating is coming off of the equipment in different formats. And so like munging it together is hard. And
yeah, so like the kind of typically you'll have a primary assay, which is telling you, does this work? Does this do the unique thing that we care about? It could be like a binding assay is a classical. It could be like a phenotypic screen or some other kind of sequencing-based assay. And then you'll often have secondary screens where you say, okay, we know that this does the thing we want, but is it toxic? Can we run another screen that kind of validates that it actually did what we wanted? So these are going to be coming out of different instruments using different kind of biological models. And yeah, so they often end up in different formats on different people's laptops or special folders.
And so the workflow on each of these pieces of equipment that drives the assay has like a GUI that allows you to export your one experiment. Right. And so experiment by experiment, the biologists are doing this analysis, but then you've got this data team that wants to look at things at a higher level, and also a leadership team that wants to look at things at a higher level.
Yep, exactly.
Interesting. I think the starting out from that place and getting to a place where you have these repeatable data processes that are producing consistent data without getting in the way of the biologists that are trying to do the real important research work of the organization is like a fundamental trade-off that I've seen you write about before. How do you navigate that?
Yeah, the way I've written about that has actually evolved as I've gone from working with Series B and C startups to spending more time talking to very early, seed stage. And because So if you're a Series B, C startup and your goal is to scale and you want to basically run as many assays as you can as quickly as possible, you really need to make sure that's done in a consistent way so that you're not spending extra time going back and cleaning things up. If you're at a very early stage and you've maybe run that assay 2 or 3 times over the course of many months, putting in those processes is actually more investment than just cleaning them up as you go along. So I think it does really depend on the rate at which you're collecting data and the rate at which you expect to collect data in the future. The important part is that you have a kind of standard that where it ends up is in a clean, consistent place. And if it takes a lot of manual work to get there, that's fine. Once you have that in place and you start to get to a point where that cleanup is starting to be a pain, that's when I think you start thinking about, okay, how do we automate that? How do we go upstream and make sure that the scientists are running the assay in a way that it's easy for us to get it into the right form? And then, of course, hopefully by then you've built up enough goodwill with the bench scientists that they're willing to make those changes because they can see the value downstream.
Yeah, I'm imagining that there's also sort of what's the level of organizational urgency around this particular problem versus the problem of just finding the targets that you want to go after as an organization. Is it the idea that there's a funding round that the startup needs to go after that's driving the urgency around bringing the data together, or where does that come from?
Yeah, I think it does coincide to some extent with the funding rounds. And of course, depending on the market, the expectations at each round may change. Maybe in 6 months, you'll have to adjust the expectations accordingly. But typically, if you're in that seed stage where you don't really know if the science works yet, you don't need to put too much thought into this, because either it's not going to work and you're going to throw everything away and move on to the next idea, or if it does work, you're going to want to validate all of those experiments with better assays. And so you're going to throw away the data anyway. So at that point, it's fine to be a little bit more disorganized. And so it's really when you get to Series B that I think most companies, they want to ramp up screening, ramp up their experiments. Then now it's really not worth putting that time in, and it's better to invest in an automated pipeline.
Yeah, and this conversation takes me back to what you were saying earlier, that ideally you would be there in the room before all of these decisions, decisions that have organizational momentum are made, because you can help organizations to do a lot more with a lot less if you're there at the beginning to help them design how they're going to collect and organize the data, versus having all of the data lying around and having to collect and organize it and automate it later on.
That's right. Yeah. I think the longer you wait, the harder it is to go back and clean it up, as with many things.
If you wake up one morning and realize that you need to raise another round, you're in a much more difficult position if you didn't do that strategic work up front.
Yeah, both when you start building the data room for your investors, but also I think there's more and more VCs who are going to start asking about, like, thinking of your data as an asset.
Yeah, that makes a lot of sense. I know that one of the things you focus on is the way that cultural components and like the different stakeholder groups that you have in biotech organizations contribute to the sort of systems and process problems that we see. Can you just talk a little bit about what you see on the ground in biotech organizations?
So I think one of the things that I've found most interesting is when I come in from living in the physical world where there are rules of physics and they almost always work. And if they don't work, then you get really surprised. Biologists typically live in the world of biology where the rules are really more suggestions, right? The exceptions are more common than the things that follow the rules. And when I think of an experiment protocol, my kind of inclination is to think of it as a series of steps that you kind of swap things in and out, and they're all going to work, and it's just going to work, right? Whereas biologists, when they see an experimental protocol, they think step 1 isn't going to work, and here's 3 reasons why. And if you're going to swap out, if you're going to make changes to this protocol, well, you're going to have to make all these other changes to the steps for that cell line, for that compound, for whatever it is, to make that work. So biologists tend to think in terms of exceptions, and they see each experiment as a unique snowflake because it is. Whereas the data folks tend to assume that it's all just templates that you can swap things in and out of. And so I think that is at the root of a lot of the clashes. It's the way that people communicate, right? It's very rare to look into your kind of motivations and your underlying assumptions to the point where you can dig down to those differences. And often you may not even realize that those differences exist until you come back 2 weeks later and you realize that the thing that you thought you had agreed to, they thought you had agreed to something completely different, even though you both agreed to it at the same time. The more you have these conversations, you can find ways of teasing out those differences and kind of change your expectations accordingly.
Yeah, interesting. So from the data team's perspective, they're just trying to get at the metadata, right? Let's have a bunch of columns that we're going to fill out for every experiment. And every single experiment is going to have these columns filled out with consistent values that we can use to build our models, do these analysis, produce these tables. And from the biologist's perspective, the columns, the fields are changing every single experiment.
Yeah, exactly.
And also you're like constraining their creativity about the experimentation process by forcing them into this.
Right.
Is that right?
Yes, no, exactly. And so the biologist's view of an experiment is this kind of process view of here are these very detailed steps that we went through in order to make the experiment work. And of course, someone who knows how to interpret that can extract those columns, those values that the data scientist needs, right? But the data scientist isn't necessarily going to feel confident doing that. And the biologists, they've gone through that process. They've gotten what they needed out of it. They don't necessarily want to go back and pull that all out because it's, for them, it may not be worth their time. And so you end up with this kind of translation problem of this detailed process view into that column view that you described. And right now, the tools available don't make that easy to do. And it's unclear if there actually will be that tool.
Yeah, that tool that spans all of the biotech R&D use cases and all of the data uses, a high bar to ask for. It strikes me that the things that make biologists good at their work, which is like the ability to follow these complex path-dependent processes through like convoluted and hard to discern patterns, also make it difficult for them to see the world from the data team's perspective. But the data team's desire to organize everything and look at everything globally and just have everything fit neatly into the way that it's supposed to work for the system to produce the result that you want. It's fundamental to the type of work that these people do.
Absolutely. Right? Yeah, that's right. And I think it's easy when you're in the midst of it to think, oh, those guys are wrong, whichever side you're on, and get very frustrated. But ultimately, I think you hit the nail on the head. It's fundamental to the sort of personality, the sort of mind frame you need to have to do your individual job is very different depending on which side you're on.
Yeah. So you mentioned the idea that a tool can solve this problem. And also, I think that in the tech world, for the technologically inclined, we tend to lead toward what are the tools that we can bring in that sort of resolve these tensions for us without us having to deal with the real people problems that exist on the ground. To what extent is the data tool ecosystem enabling the process, and to what extent is the specialization of these data tools in biotech like hindering the process of getting these biotechs up to scale?
Yeah, great question. I think there's been a lot of startups recently that have been founded. I think the software has evolved from 10 years ago. A lot of what was important was like ELNs and LIMS types of systems that were solving the problems that you had today. Now, as we're getting into kind of more AI/ML use cases, the types of tools that we need are evolving. And so we're seeing a lot of startups that are trying to build these tools. I think one tendency is to try and make them broad and have a lot of functionality because it's very difficult right now to put together different modular pieces, which is the ideal, the dream that everyone has. But like those modular pieces don't exist. There isn't that standard yet. I think a lot of the startups tend to try to build the everything platform, which from one perspective makes sense because that way the individual startups don't have to worry about integration. But in practice, often the components that they need is just some fraction of that. And so it ends up unintentionally creating this sort of sense of lock-in, which is, again, I think their goal is not lock-in. They're trying to make it so that they're doing good for everyone. I think they're doing that as well as they can. But yeah, so I think we're not yet to a point where we have these kind of modular pieces that we can put together. I'm hoping that we can continue to evolve there just so that there's less overhead involved in bringing in a big solution, and it's easier to iteratively bring in small solutions that kind of add to the overall solution.
From my perspective, there's this basket of modular tools that have been put together in consistent patterns across industries. So you've got your storage layer where everybody's landing their data in S3 or Google Cloud Storage. And then you've got your compute layer where maybe it's a data warehouse, maybe it's something like Databricks, or maybe it's more of a streaming type system. And then you've got your output layer where you're doing sort of visualization and or driving value from the data. And I'm just wondering, is the biotech ecosystem recreating these tools for biotech? And where are those tools that are like at scale in the data ecosystem falling short of the use cases in biotech?
I think a lot of the components from the modern tech stack make a lot of sense. Like certainly like cloud storage, is generic enough that like you don't need to make a specific version of that. You could argue that like something like Ignite, where there are some features that are designed for sharing data with CROs, things like that can make sense, but it's more kind of on the periphery. So I think there are a lot of components from the modern tech stack that make sense. there's specific aspects of biotech data, particularly the early types of assays that startups tend to run that are different from if you're making another social network or like a e-commerce site, right? Something like that. So things like Snowflake, where it's an at-scale relational database. And I think that often makes less sense for these early stage assays where there's fewer references between tables. It tends to be more, each table is self-contained, relatively small, usually small enough that you can either put it in a relational database if you really want to, or just leave it as a file in your cloud storage somewhere. So I think a lot of these heavyweight solutions that have become the standard in the modern tech stack don't make as much sense. On the other hand, I think there's also usually the things that make these tools specific to biotech are on the periphery, right? If you have a visualization tool that can automatically draw compound diagrams from a smile string, that's huge, right? It's small. Someone could add it in a day, but like for the end user, being able to do that versus having a generic BI tool that can't do that is a big deal, right? And then you have other software that like isn't actually need to be specific to biotech, but for commercial reasons, it makes sense, where like Quilt Data, which is another great tool that started off in the tech space, realized that like most of the adoption was in bio and has kind of doubled down on that and started to like really build the features that their biotech customers are looking for, which again may not be specific to bio, but it works, and it ended up being a really good strategy for them.
Interesting. So there's a variety of things in there that I would just want to unpack. So there's the file format issue that up front, the formats of these data are not formats that are commonly used across industries. Mm-hmm. And then there's also the computational processes that are being done over those file formats are atypical computational processes, so they're not typically built in to a lot of the at-scale data warehouses, for example, are not going to allow you to do complex genomics analysis within that environment.
Right.
And then like the last thing I heard was like the outputs at the output layer, you've got data visualizations that are, unique to the type of analysis that's being done. So there's this neat connection between each slice of file format, computational approach, and visual output, that there's just lots of these that get stacked together. Is that right?
Yeah, that's fair. Let's start with kind of the contrast is the if you're building like a social network or e-commerce site, a lot of the times you have tables that need to connect to each other, right? So like if it's people versus like topics that they care about versus their connections. And so on the one hand, if you want to scale to Facebook scale, you need to do that for billions of people. You need to know all these connections. And so you need a database that will allow you to do that, right? So again, both the connections between tables and then the scale. So if you're looking at especially early stage biotech startups, the data that you can generate is fairly limited. And so if you're doing sequencing, maybe you can do hundreds of thousands or millions. But like typically, once you get down to like compounds that we care about, it's going to be in the hundreds, maybe in the thousands. It's at a scale where you don't need a huge hammer to manage data at that scale, right? It can fit in memory in your laptop. And even for when I've worked with like single cell data sets before, like even those data sets, they can fit in memory in your laptop. So having a large scale solution that's going to work for billions of users, billions of data points for a social network is just way overkill for that. Each data set tends to be, it's compounds, maybe cell line, and then numbers, right? So like the structure is very simple. You don't have these complex connections between tables. Now, once you get into like clinical real world evidence, then you start to get that. But if you're talking about early stage research, it tends to be much simpler. So like having a table saved in a file somewhere in your cloud storage is, especially when it's a handful of developers, handful of data scientists, that's usually sufficient. And it's only once you get into much more data, much more complex data, that you have to worry about these other solutions. So even then, these kind of massive cloud data warehouses is often still overkill.
Yeah. So what does that architectural pattern look like then when you're moving from like you're doing this exploratory research where you've got a team of scientists who are working on their laptop with files that all fit in memory to, you've got these repeatable analyses that you want to do over and over again? Is it basically just Dockerize the thing that you were doing on your computer and then run it over a bunch of files in cloud storage, or how does it go?
Yeah, I think certainly that can work up to a certain scale, right? At some point you'll need to introduce some kind of proper database. And usually, again, that's when you want to start looking at assays with each other. So like files work if you're just updating it a few times a week or maybe even every few weeks. You can just rewrite the whole thing. It's once you start getting enough updates that you don't want to blow up and recreate your table every time you do the analysis. That's when a database allows you to do that incrementally, and that it's worth that investment. But depending on how fast you're moving. And of course, if you're a platform company, the calculus is very different because you know you're going to get to that stage. And so you might start building that sooner. But yeah, that's roughly the calculus.
Are you a biotechnology company looking to unlock the potential of your business data? CorrDyn can help. We're an enterprise data specialist that helps companies working in life sciences make smarter strategic decisions. From developing the right data strategy that starts with our data maturity assessment to building and delivering bespoke technical solutions, we are equipped to tackle the most complex data challenges. We have partnered with dozens of high-growth organizations, from manufacturers of custom oligonucleotides to molecular diagnostic companies, to achieve data competency. Whether you need to supplement existing technology teams with specialist expertise or launch a data program that lays the groundwork for future internal hires, you can partner with CorrDyn to unlock the potential of your business data today. Simply visit connect.corrDyn.com/biotech to learn more. Now, back to the show.
I want to play a little game of fact or fiction. This is the first time we've ever done this on the Data and Biotech podcast. So I'm just going to throw some ideas at you that I hear in the biotech data ecosystem and get your takes on them, if that's all right.
Yeah.
So the first one is that AI and software are going to automate most biotech R&D processes within 10 years. So is that fact or fiction?
I try not to make predictions about the future, or at least not publicly. I think there's what's going to come before the AI and data is going to be automation. People who know more about automation than me have predicted that most of the labs are going to be automated in 5 years, which maybe that's optimistic. Certainly, they would probably say in 10 years. And I think once you have lab automation, it's much easier to automate the data because this metadata that you mentioned earlier, that if a robot is picking out the plates and all that, you can extract that all from the logs, where it's much harder to extract that from the mind of a person who was sat there with a pipette. And so I think if lab automation is the standard in 10 years, then I think getting that relatively clean data into an automated pipeline is much easier than, and so I think it greatly increases the chances of that automation.
So the path to the data ecosystem that we're looking for is a path of automating a lot of the research processes up front so that the data flows naturally in a format that is easily consumable by the data systems that data teams will tend to manage.
Yep.
Okay, interesting. Data is more important than machine learning models in biotech.
Yeah, so I think that's true. And if you look back at machine vision, image recognition, the convolutional neural networks that are now the standard for doing this, or at least maybe they've been replaced now, but for a while they were the standard. Those existed since the 80s, at least on paper, but there wasn't enough data to actually make them more usable, more effective than other methods. And so it was only when you got ImageNet with millions of images that those models that had been around for decades suddenly started outperforming other models. And I've seen this a lot with biological data sets, which are currently much more constrained than the kinds of data sets in the tech world, where you basically get to a point where even very simple machine learning models do as well as the very sophisticated ones, sometimes even better, because you've reached a constraint on the data. And so I think in general, models are fun, but if you don't have the data, they don't work.
The next one, fact or fiction. Biotech is a useful industry descriptor for vendors that are building software and data apps to target as a unit.
I think that's true, because even if the software is underlying mostly the same, I think for a vendor to understand the problems and understand the terminology well enough to be able to tell you how their solution fits in, I think that's really important. So yeah, the software itself may not be that different, but I think it is a valuable distinction.
Interesting. What made me think of that one was the idea that a lot of the work that these biotech organizations are doing is very different depending on the types of biological processes that they're working with, the types of assays that they're using, the types of target, the biological specializations that they're operating in. But it sounds like you, based on your experience, view the problems of these very different biotech organizations as being relatively consistent.
So on that side of, I guess if you're comparing it to like general for software for any industry, I think it's a more useful category than is the industry in general. I think once you get into individual biotechs, you find that there actually is a lot of heterogeneity between what different biotechs need, which is a lot of the reason that you don't have any. It's very hard to build a tool that serves a lot of different biotechs and is very specific. And so I think every company that's building software for biotech has to make that decision of, do we go deep, and build something that's really useful for a very small number of potential clients, or do we go wide and build something that's slightly better than generic?
Yeah, that makes a lot of sense. So there's the micro-targeting approach where you sort of get deep with a particular segment within biotech or within a particular problem space, but then there's also the broad approach that you can take as well. So the last one we'll play with fact and fiction is fact or fiction. Hardware and software vendors in biotech are more interested in locking data in than enabling analysis of it.
Yeah, so I have to be careful because a lot of these are folks that I want to work with. So in my experience, I think it's a mix, right? I think hardware vendors are more likely to try and lock in data because investors are telling them that hardware is not a great investment. And if they can build software and become a software play, then their valuation will go up. And so in order to become a software play, the best way to do that is to lock your clients into your software system. And so I'm not going to name names, but I do think that is a motivation for hardware companies. For software companies, I think it's much less common. And in fact, I can't think of any software companies that are intentionally trying to lock people in, and especially the newer companies are really great about saying, we're going to use whatever. We'll at least give you the data in a way that you can pull it out of the system, or we'll let you use it in your own cloud, wherever you want it. And that's because I think they first of all don't want to have customers be afraid of lock-in. They know they'll get more customers by telling them they can go anytime and keep them longer, because if they tell them they can go they're already doing that software play. So if they can give more value, then they think they can do best that way. So I guess so I think some hardware companies, not all hardware companies, maybe one or two software companies, but not most of them.
Awesome. Thank you for experimenting with fact or fiction with me. I enjoyed your responses. I noticed that you started on a project called Foam for data apps in biotech. Could you just talk a little bit about that project and why you started it up?
Yeah, so this was kind of part of this effort that I mentioned earlier of trying to find a way to make more modular components for building data and apps. And I think one of the things that I found was that if you want to build analysis pipelines, that's pretty easy. But when it comes to collecting data from users, that's where it gets tricky, because ELNs and LIMS are not really set up for AI and ML applications. And so if I just want to let users enter sample IDs and give me some metadata about the samples, there isn't really an easy way to just spin up that app and have that be integrated. So the idea of Foam is it's what I call low code for coders. There are a lot of these low code app development frameworks where you go in with a mouse and you can draw the windows that you want, and then you design the underlying schema, and then you click go and you have that app. I think that's great as a way of quickly spinning up these apps. But the problem is that if you're a coder, it's awful because you want to work with your keyboard. You want to be able to copy and paste. If you've got to do the same thing a dozen times, you want to be able to use your fancy IDE magic to do that. And so the idea of Foam is that it's similarly configurable, except that it's all based on YAML files. So YAML it's like a version of JSON. It lets you write these sort of structured configs. And so the idea is in a text editor, you very concisely define what you want the backend schema to look like and what you want the different windows in the workflow to look like. And then in a CLI, you say compile, and now it generates the code using Django for the back end and React for the front end. So all fairly standard open source tools. And then that code you can run locally, you can put it in a Docker, you can push it to GitHub, you can do whatever you want. So it's meant to be very developer friendly, but allow developers to very quickly spin up these front end applications. Maybe even if you're a back end developer and don't want to write React code, you can write one of these configs and get your app going. It's in very early stages. It's a lot of kind of cardboard and duct tape. It's at a point where it was enough to convince me that it works, and I'm planning to put a lot of energy over the next months, maybe years, to get it to to a good place. And if anyone wants to try it out and see if it works for your startup, send me an email. I'd be happy to, for free, jump in and give you some advice and help you work through any issues.
Is the ideal application for foam a data entry application that keeps track of the meta, as we were talking about earlier, or is it like what do you expect to be the end user problems that you would solve with apps that are built using foam?
Yeah, great question. I think I'm probably being very ambitious, but I think you can start off as data entry. It's designed in a way where you can start building tables that you can use to get insights into what's going on and maybe even share those with leadership. So it's sort of anything that you can build with data entry forms and then tables. And then if you want to build custom React code to build very custom views, it also allows you to insert that. So I think of it as like filling in, the reason it's called foam is it's meant to fill in the cracks between the other pieces of software that you have. And so I think ultimately, I'd like it to get to a point where you could build the kind of the core infrastructure, core data management infrastructure for your whole organization. Maybe it someday could replace the ELN, maybe replace the LIMS. Definitely not today, but yeah, I'd like it to be able to do anything that involves either entering, finding, or looking up data.
Yeah, so it's just interesting to me that you took this approach to solving that problem based on your experience with biotech startups, because it strikes me that it's an approach that has a lot of respect for the diversity of like issues in the data ecosystem from company to company and the diversity of data sources and interfaces that companies are working with. And so like foam is like filling in the gaps of whatever the other tools are that are on the table.
Yes, that's the goal. And it's still very much an experiment. I don't know if this will actually work, but I figured it doesn't hurt to try. And it's something that I had been thinking about and had built other versions of enough times that I knew at some point I was going to need it again. So may as well build it open source.
I think the idea that this is the solution that came to you from your experience is interesting to me as the actual code that's written to solve the problem in this way, using Django and React. So that's cool. An idea that I've heard you tossing around is this idea of a digital twin for the lab environment. What does a digital twin of the lab environment mean to you?
Yeah, so for me, what it means is it means that I want to know where everything in the lab is at any given time. And I don't just mean like in terms of inventory, but also in terms of like, what are the experiments that people are in very early stage discussions about, all the way through the ones that are in the lab or the ones that have come out already. So I want to be able to look and see, like, for any question I have about the experiment flow, the lab, the pipeline, any of that, I want to be able to query the database and find that answer. So very operational. The problem that you often run into is data scientists on a digital team in a biotech. Every once in a while they get an email, hey, did you analyze that data yet? And they're like, what data? I don't know what you're talking about. Okay, because we need it in an hour, right? And for the data scientists to be able to know when the data is going to come off the machine so that they know if they have time in their schedule to analyze it, whatever it is, like all those questions all the way back to the very early ideation. I want that all in a database.
Yeah, interesting. It's like a combination of the metadata of every experiment, every input, every output, but also bringing data science into the production process of the research that's happening, allowing them to integrate more seamlessly into it by understanding everything that's happening.
Yeah.
Other than just a database that has all of it in there, what would that look like to you?
I think what it would mean is having software that helps the wet lab, the bench teams do what they need to do in a way that it's recording kind of all of those steps as you're going along. So once they say, oh, I really want to answer this question about these compounds, they can go into the tool and it encourages them to say, okay, here's an experiment that I want to run. I don't know when I'm going to run it, but I'm just going to put a placeholder there. And then as they flesh out that experiment, it makes it easier for them to define that. And so again, now you've got records of all these experiments that may happen. And the data scientists maybe can go in and say, oh, if you change these parameters, it's going to be better in the end. And then continue to facilitate that process all the way down to where they've put the protocol together, they put all their plate maps together, and it's ready to go into the machines. So I think having software that encourages them to use the software and helps them, give them an incentive to like record all of that.
What I hear you talking about is like the collaborative elements of it, of both giving the data team and scientists in the wet lab formative input into what's happening, but then also ensuring that both parties are getting what they need from that, like the initial phase, and what they need from the outputs, beginning with the end in mind
Yes, absolutely. Yeah. Because the only way that you're going to get users to do this, like to put in the effort that make sure that data is there, is if they're getting something out of it. If they can enter the experiment details in a way that allows a robot to do the experiment for them, maybe that's the incentive. Maybe it's something else.
It wouldn't be a Data and Biotech podcast if we didn't at least touch on AI and ML. What role do you see AI and ML playing in the biotech startup space right now, and how do you expect to see that role evolve?
Yeah, it's starting to be everywhere, right? From target discovery to interpreting results from instruments, so particularly like for high content imaging, HCI, right? Like phenotypic discovery. I think there's a lot of really interesting stuff there. It's coming in analyzing sequencing data, looking at NGS data, things like that. Yeah, I think we're already seeing it being applied pretty much every place in the pipeline. I don't think that's going away with like ChatGPT, LLMs, things like that. Harder to see where that fits in. I think the way I see those tools being used, or the way I would imagine them being used, is as more almost as a learning tool, because you want to check what they do, right? So if, say, like you're designing an experiment, right, you could maybe type in some free-form text that says, here's how I want you to do the protocol, and maybe it writes it out for you, but you're still going to want to check that, right? And at some point, writing it out long form is going to be more effort than just going in and designing the details. So I think as a bridge to those kind of more specialized, and maybe beyond that too, but that's my current thinking, is that's where there's a pretty good use for it, need for it.
Yeah, that makes a lot of sense. So there's the traditional machine learning that's being applied at every phase of the drug development process, and then there's LLMs that are helping to reduce the manual effort of generating these long form pieces of content, but also try to learn what best practice is and embody that best practice in the way that you're applying it there.
Yeah, that's right. Yeah. And we've already seen some POCs of this. I know Synthase a few weeks ago, maybe months ago now, released a blog post about this. And then a couple of others have as well.
Well, as we bring the conversation to a close, I'm just curious if there are any pieces of content or anything that you've listened to or read recently that you think has been really interesting or helped to understand the space a little bit better.
Oh, that's a great question. So Kaleidoscope just put out a blog post on the different phases that biotech startups go through, where they asked VCs, what are you looking for at each phase of that? That was really interesting, especially the parts about data assets and how important that was. And then there's this guy, Ben Stansel, who has a newsletter about the kind of modern data stack in general. And he had this really interesting one just a week or two ago about why kind of big solutions are often hard to adopt and why corporations often only need 10% of this, kind of the big solution. I thought that was really interesting as well, not for biotech, but in terms of the question you had asked about kind of software adoption and what are companies doing right or wrong. Yeah,
interesting. Jesse, I really appreciate you joining us today. We'll post all the places that people can find you on the blog post that we associate with this, so we don't need you to say it online. But thanks so much for joining. It's been a pleasure, and we'll talk down the line.
Thanks so much for having me. This was a lot of fun.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.